dobry Państwu. Miło Was gościć na ostatnim wykładzie dzisiejszego dnia. Mimo poprzednich trudności technicznych, naszym gościem jest David Nutt, profesor neuropsychofarmakologii w Imperial College w Londynie. Był m.in. kierownikiem oddziału badawczego w Narodowym Instytucie Nadużywania Alkoholu i Alkoholizmu w USA, przewodniczył Europejskiemu Kolegium Neuropsychofarmakologii, czy też założył organizację Drug Science. Wkład profesora Nutta w farmakologię obejmuje badania nad funkcją receptora GABA i noradrenaliny w zaburzeniach lękowych, funkcję serotoniny w depresji, funkcję endorfin i dopaminy w uzależnieniu oraz użyteczność neuronaukową i kliniczną psychodelików. Ma na swoim koncie m.in. ponad 500 prac naukowych oraz 36 książek. Za moment wysłuchamy jego wykładu The Psychedelic Revolution in Neuroscience and Psychiatry. Zachęcamy do aktywnego uczestnictwa poprzez zadawanie pytań zarówno na czacie na YouTubie, jak i na platformie e-kursy. Odczytamy je naszemu gościowi tuż po wykładzie. Życzymy miłego słuchania. Now in English. Welcome, ladies and gentlemen. We're happy to have you here. We're glad to introduce today's final guest, Professor David Nutt. He is the professor of neuropsychopharmacology at Imperial College in London. He was inter alia head of the research ward at National Institute of Alcohol Abuse and Alcoholism in the USA. He has held presidencies including ECNP and founded the charity Drug Science. Professor's contributions to pharmacology include GABA and noradrenaline receptor function in anxiety disorders, serotonin function in depression, endorphin and dopamine function in addiction and neuroscience, and clinical utility of psychedelics. He has, among others, over 500 original research papers and 36 books. Now, Professor David Nutt will give a lecture entitled The Psychedelic Revolution in Neuroscience and Psychiatry. One information to the audience. We invite you to ask questions by YouTube chat. Enjoy listening. And Professor, the floor is yours.
Thank you very much, Alex. Well, it's a pleasure to be talking to you. Apologize, it's later than planned, but there was a mix-up with the times. But at least we're here and we're all here, I hope, so we're all going together. So when I talk about psychedelics, what am I talking about? Well, here are some examples. I'm largely going to be talking about substances which emerge in nature that have effects on the brain. And in this slide, you see some of the more well-known ones. The top left is mescaline, which comes from the peyote cactus. Then you have psilocybin, which comes from magic mushrooms, of which there are over 200 species in the world. Ayahuasca is a cocktail of DMT, dimethyltryptamine, which is not normally orally active, but over the millennia, the indigenous peoples of the Amazon basin have worked out that if you brew it with another plant that produces a substance called harmaline, it makes the DMT available orally, and that's because the harmaline is a monoamine oxidase inhibitor, and it prevents the breakdown of DMT. So that's ayahuasca is a drinkable form of DMT. And the top three, they're all serotonergic psychedelics. Bottom right is the Northern European psychedelic mushroom. It grows in many countries in Northern Europe, including Poland. And that's the Amanita muscaris. And that's a different kind of psychedelic because that's a GABAergic psychedelic, but I won't be talking more about that today. But I just show this, it's been around for quite a few thousand years. There's an old Roman mosaic, you see, showing how to make the mushroom tea. Throw some mushrooms into a pot of hot water. And then back to the serotonergic ones, there's morning glory and there's ergot. And these plants make ergot derivatives, which are the nearest things in nature actually to LSD. And their psychoactivity was long known. And the most important image here really is the bottom left-hand image. That's a Greek vase. It's 3,000 years old. And it shows the Greek god Demeter partaking of ergot. And the Greeks knew that if they ate ergot when it grew on the rye and drank wine, the cocktail was very powerful, and they celebrated the emergence of the ergot with their holiday seasons. They went into the countryside and they had music, they had it was like a big party, like a big rave, music, theater, dance. They called them the mysteries. And from those experiences, they came back and did the things that we usually associate the ancient Greeks with, such as geometry and philosophy and logic, and of course the creation of democracy. And there is little doubt that Western society is largely embedded in the constructs that emerged from ancient Greece, and it's therefore quite plausible that the psychedelic effects, or the mind-opening effects of ergot, may have been the underpinning driver for the evolution of our societies. And of course, if you go to other societies like Hinduism, you discover that other drugs, possibly mushrooms and ephedra and cannabis, were equally important in much of their cultural development. Moving on to the beginnings of modern neuroscience, and here's one of the founding fathers of the discipline of psychology, William James. And having taken mescaline and having taken nitrous oxide, he wrote about his insights in this way. He said, our normal waking consciousness is but one special type of consciousness, whilst all about it, parted from it by the filmiest of screens, there lie potential forms of consciousness entirely different. And the next statement I've put in bold because it really is a challenge to all science. No account of the universe in its totality can be final that leaves these disregarded. And he's saying essentially the study of how drugs change consciousness is one of the fundamental questions of science. But in his day, it was difficult to study them. How to regard them is the question, for they're so discontinuous with ordinary consciousness. And now, over 100 years later, with modern imaging, we can study them. Perhaps the most famous protagonist of the need or the value, sorry, of perhaps of taking psychedelics was Aldous Huxley. And his book, The Doors of Perception, has become very famous. But what most people don't realize is that the title, The Doors of Perception, was taken from the writings of an English mystic, painter, a sort of philosopher called William Blake. And Blake said this, as the doors of perception were cleansed, everything would appear to man as it is, infinite. For man has closed himself up till he sees all things through narrow chinks of his cavern. And what Blake was saying was that humans don't use their full capacity of their mind and their vision and their thoughts. They basically have a very limited, closed approach to everything. And the reason Huxley used the doors of perception, because he realized that mescaline and subsequently LSD essentially cleansed the doors of perception. They opened his mind. And then he thought, well, hang on, if my mind is being opened by these drugs, what is closing my mind? And he came to this wonderful conclusion that it was the brain. And he said the brain is an instrument for focusing the mind. And one of the most pleasing things about our research is that we've actually proved him right. About the time Huxley was writing The Doors of Perception, this man, Albert Hofmann, was making available through his company Sandow this more potent derivative of the ergot psychedelics, LSD. And his description of his accidentally taking it and the normal 30-minute cycle ride home, seeming to take 7 hours, is now legend. There's even a day, bicycle day, named after it. It's the 20th of April. And he was a great supporter and a protagonist for the use of LSD. He took it on a regular basis in what you might call MIDI doses. He lived to over 100. There he is on his 100th birthday in his psychedelically colored house overlooking Lake Geneva. The first British psychiatrist to take it was, he lived to 103, having taken it in 1953. So the idea that these drugs have a deleterious effect on your brain and your functioning probably already disproven from the very start. But of course, the idea that these drugs are harmful has been developed and exaggerated over in many years in an attempt to get them banned. But before the ban, they were widely used. They were seen as a remarkable advance. Remember, in the 1950s, we didn't have antipsychotics, we didn't have antidepressants. All we could do with people who were mentally ill was sedate them with opium or with barbiturates. And psychedelics like LSD here in the box, and subsequently psilocybin, which Hoffman discovered was another psychedelic with a similar chemical structure to LSD, and which was made and sold as a medicine by Sandow in the 1950s and 60s. They were widely used because they offered treatments for psychiatry that we didn't have up to that point. And they were used for 4 different purposes. One was to model psychosis, to try to understand the nature of the brain changes in psychosis. And we're beginning to do that today now. We're using psilocybin in healthy volunteers as a test bed for new non-dopaminergic antipsychotic drugs. And one of them we discovered is a tyrosine kinase inhibitor called saracatinib, which attenuates the effects of psilocybin, and which we're now testing in early onset schizophrenia to see if it might have an antipsychotic effect. And it was used by mental health professionals to understand the nature of altered consciousness, and it was often given to junior psychiatrists early in their training so that they could appreciate what it might be like to be a patient. They could understand that their brain could be changed in a way which could be quite challenging. And then it was used in therapy, and there were 2 kinds of therapy. There was psychedelic psychotherapy, which was largely favored in America, and that's mostly what we do today. We give 1 big dose, people have a powerful experience, a trip, and they often come out of that changed in a very positive way. And then there's also other technique is called psycholytic psychotherapy, and this is giving lower doses, not microdoses, but what we call a MIDI dose. And the MIDI dose, you can detect an effect, but you don't hallucinate. And that was given before each psychotherapy session so that people would engage better in the psychotherapy. It would break down their resistance to the analytical techniques that were being used at the time. And we're doing that now in a study with OCD patients who don't want to have a full trip, but are prepared to take a lower dose to see if that can help them engage better in their behavior therapy. Sandow made the drugs available worldwide. NIH in America funded up to 140 grants. So there was an enormous amount of research, 1,000 clinical papers, 40,000 patients, 40 books, 6 international conferences. And in 1971, coincident with the banning of these drugs by the international UN conventions, Masters and Houston pulled together all this data and came to the conclusion that the results were overwhelmingly positive, describing safe and effective treatments. And here's just a snapshot of some of the major papers which came out. I'm not going to go through them in any detail, but you can see they've got large numbers of subjects, 5,000 subjects, 4,000 patients, 50,000 drug-assisted sessions. And what's remarkable about these huge databases is the very low level of psychosis and the very, very low level of suicide, probably lower than you'd expect from this population if they weren't being treated. So at the time, this was seen as a revolution, and there was a great hope that this would not only transform psychiatry, but also transform clinical neuroscience. And summing it all up is this statement here: treatment with LSD is not without acute adverse reactions. Of course, some people can have tough trips, but given adequate psychiatric supervision and proper conditions for its administration, the incidence of such reactions is not great. But in 1967 in the US, in 1971 in the UN, these drugs got banned. And the impact was absolutely terrible. So what you see here are the number of publications each year in the y-axis for LSD in blue and psilocybin in red. And this big increase up to the late 1960s as the US government particularly was funding a lot of research. And then the bans came in, and after a couple of years, people publishing stuff they'd already done, research virtually disappeared. And there were 2 reasons for that. The first is the US government, which is the major funder of biomedical research in the world, has not funded any studies since. And secondly, if you could get money to do this research from charities, for instance, or philanthropists, you couldn't get the drug because the UN conventions were interpreted by almost every country in the world as that these drugs were too dangerous to research. And there was a deliberate attempt to try to eliminate all knowledge of psychedelics. The hope was they'd be forgotten about. And thankfully, you can see the pictures beginning to change at the end, and now we're fighting back because these truly are remarkable drugs with remarkable potential. If you want to read more about the hysteria that led to the bans, you can read these reviews of mine, which not just cover psychedelics, they also cover cannabis and ketamine as well, and MDMA. The banning of these drugs is the worst censorship of research in the history of the world. It's truly outrageous, and it's lasted over 50 years now. Well, about 15 years ago, my PhD student, Robin Carhart-Harris, and I decided it would be timely to ask the question, what is a psychedelic and what does it do? What is the brain signature of psychedelia? And we decided to start working with psilocybin. Here's another magic mushroom species, the liberty cap, which makes psilocybin. And psilocybin is in this little circle here. And this graph shows the relationship between all the serotonergic hallucinogens in terms of their affinity at the 5-HT2A receptor and their potency in humans. And the very potent drug, LSD, you give in very low amounts, microgram quantities. Psilocybin is moderate potency, you give in low milligram quantities, usually 25 milligrams. And mescaline, you give in high milligram quantities. And the relationship between affinity and potency is so tight that that proves that all these drugs work on the 5-HT2A receptor. Where are these receptors? Well, this is a heat map of them in the human brain, and you can see they're in the cortex, they're not in the midbrain and brainstem. And they're in the cortex, they're highly expressed in the posterior and anterior cingulate cortices in the frontal cortex, lower expressions in the sensory motor strip and the visual cortex. And these regions where there's the highest density of these receptors are the regions, of course, which are the most recently evolved parts of the brain. They're the parts of the brain which are biggest in humans. Some people think that it's the receptors themselves which have driven this massive expansion of the human brain. But what's for certain is that the impact of psychedelics on the human brain is targeting those very high level human brain structures, which give us the unique properties of humanity, particularly constructs such as imagination, analysis, integration of complex ideas, etc. So these receptors target, as I say, the very highest levels of our cognitive function. And the receptors are particularly highly expressed on the pyramidal cells that communicate across the brain. So for those of you who don't know, the brain is essentially a kind of three-dimensional computer. The first two dimensions are linear dimensions, and this is, for instance, in the cortex. These projections from the cell bodies here in layer 5 pyramidal cells, these are part of what are called cortical columns, and there are billions of these cortical columns in the cortex, and each column is like a computer. But the communication between each of them is what makes the brain so special. So you imagine you've got billions of computers all linked together. And the linking together comes from these layer 5 pyramidal cells, which pull together the information from all across the brain, which is, so the reason the brain is more powerful than all the computers on Earth today, one human brain is because it's actually billions of computers all working in a unison using a very sophisticated and as yet not fully understood software. But the 5-HT2A receptors are densely expressed on these layer 5 pyramidal cells. And when you stimulate these receptors, you get a massive depolarization of these pyramidal cells. And that's what perturbs the brain function and leads to the altered brain signatures that we see. And I'll show you in a minute. So about 12 years ago, we did the first experiment. We took people who used psychedelics, we put them in a scanner, we gave them psilocybin. They had a range of experiences, the colored lights like Christmas tree lights, the elemental hallucinations, a sense of body morphing and disturbing and atomizing, and many of them floated out of the scanner and one went to heaven and bowed at the foot of God. They all came back. And when we got them out the scanner, we analyzed their brains, and we found, to our absolute amazement, there was nowhere in the brain where there was increased activity. And we'd expected a lot of increased activity because of these experiences. In fact, we just found decreased activity in the posterior and anterior cingulate, and also the thalamus. And this is probably secondary to the changes in the cortex. We thought we'd done the experiment wrong. That was arterial spin labeling. We went away again, and then we repeated the whole thing using fMRI bold, got exactly the same results. And then we realized we discovered something really important, that actually Timothy Leary and the psychedelic enthusiasts of the 1960s who said these drugs turned on the brain were actually wrong. They turn off the brain. And they turn off the brain in the areas where these receptors are. And that's probably because of the massive rebound inhibition that activation in the pyramidal cells causes. And this turning off was strongly correlated with the experience. So here you see turning off activity in the anterior cingulate cortex. Reduced activity was associated with increased intensity of drug effects. So that time we scratched our heads and said, well, what's going on? And we looked at the literature on these regions and realized that another way of looking at those two regions had been developed with the concept of them being connector hubs. They're parts of the brain which integrate brain function. The anterior cingulate integrates processes such as attention and concentration and memory and emotions. And the posterior cingulate integrates all the sensory inputs, the vision, the hearing, the touch, the proprioception, the taste, etc. And these are like the two parts of the brain which conduct the brain. They're not the conductors of an orchestra. And so the analogy we used was if you take away the conductor of the orchestra, then the orchestra might decide not to play Bach. It might decide to play jazz, for instance, play differently. And we think that's what's happening. Psilocybin disconnects these connector hubs in the brain and allows the brain to work differently. And it turns out that that is the case. And what was particularly pleasing about that discovery is it kind of validated the Huxley prediction. Huxley had said there's a reducing valve. The brain is a reducing valve. It limits the capacity of the mind. And in doing so, it lowers the efficiency of the brain as an instrument of focusing the mind. So he said psychedelics turn off that reducing valve. And that's, I think, what we showed they do. So here, this is how we currently conceptualize it. I'm going to use vision as the example because we know a lot about the processes of vision. We know that your brain's not a camera. We know that when you see something, photons from the image go into your retina. The retina then turns them into electrical impulses, which pass up the optic nerve through the superior colliculus into a whole range of different posterior brain regions, which reconstruct from these electrical impulses, the where, the what, the how, the movement and the color, etc., of what's out there. So your brain reconstructs an image of what it expects to see. And that's why the brain is a very efficient computer, because it doesn't bother to do real-time upgrades. It essentially makes a prediction and works on that prediction unless something significant changes. When it changes its prediction. But the brain can't do that. If you take away or disrupt the layer 5 pyramidal cells, which are vital for this reconstruction, then it doesn't happen. And what you see are these kind of elemental images. And we know from work in frogs and in other animals that the primary processing of the visual cortex, what's built up from the electrical impulses, are these elemental shapes, circles, spirals, and simple colors. So under a psychedelic, when you're hallucinating these experiences, you're actually seeing, for the first time since you were a baby, the primary workings of your visual cortex before they're built up and turned into what you see as a full image. And that in itself, I think, is really quite elegant. Now, by switching off these control centers in the brain, we allow the brain to become hyper-connected. And on the left-hand side, you see the normal state of connectivity in the brain. And each of those images are made up of 7,200 voxels in the brain. And what we're looking at is the likelihood of them changing their activity in synchrony. On the left-hand side, you see that most of the connections are around the edge, and this is called the small world brain. Visual cortex talks to visual cortex, auditory cortex talks to auditory cortex. Of course, there has to be some cross-cortical communication. If you see a bus driving down at you, then you've got to move your legs to run away. But the reason the brain is such an efficient computer, and it's 10 times more energy efficient than any human-made computer, is because it doesn't do a lot of this cross-cortical communication, which is energy expensive. But under psychedelics, the control systems are broken down. And so the brain is no longer constrained in this way that it's been forced into over decades of development and education. It can liberate itself. It can make connections it wasn't allowed to make or hadn't made since childhood. And that increased connectivity turns out to be really important in our understanding the psychological effects of these drugs, because it allows people to think differently about their past and uncover memories that have been repressed. But it also allows them to make new connections, to make sense of things in a different way, to reframe and reanalyze assumptions they've made. And then in the case of conditions like depression and addiction, come up with new solutions to the problems that have been bugging them. Having done the psilocybin experiments, we went on and did the same with LSD. And here you see a very beautiful image, explains the nature of complex hallucinations with eyes closed under LSD. Normally, if you close your eyes, you don't see very much because your visual cortex, V1 here, just connects to V2, a little bit to the hippocampus. This is the small world brain. But under LSD, the visual cortex connects to everywhere. And it's that massive hyperconnectivity which underpins the very powerful, vivid, and often very entertaining and interesting hallucinations that people have under LSD. It also helps to explain the phenomenon of synesthesia, which can be exaggerated by psychedelics too. Getting this stuff published was quite challenging. Referees were rather cynical. One said it's all just blood flow changes from 5-HT receptors on blood vessels, which of course it couldn't be because, well, A, it didn't happen across all the brain, but B, even if that were true, that would still be a very powerful explanation of the psychedelic experience. What do you expect if you mess with the brain with psychedelics? Well, I'd expect slightly better refereeing, frankly. And this highlights a very important problem with this research, is that people don't want to know what psychedelics do because they've been vilifying them and criticizing them and saying they're harmful for so long. And there's a wonderful quote from Huxley again, which sums it up, even though this was about politics rather than about science. But all you scientists here should remember this and stick it up above your desk. Orthodoxy is the die-hard of the world of thought. It learns not, neither can it forget. And psychedelics are redefining many aspects of neuroscience. They're breaking the orthodoxy. And a lot of people don't like that. However, we listened to the referees' criticisms, we went away, we repeated the psilocybin experiments, but this time we measured the effects in the brain using magnetoencephalography, which, as you probably know, measures the magnetic changes produced by electrical activity in the brain, so we could measure directly electrical activity. And we found the same thing. We found this profound disruption of synchronized activity but across the whole frequency range, particularly in the posterior cingulate cortex, but also at this higher frequency range in the anterior cingulate as well. And the posterior cingulate changes correlated beautifully with the sense of ego dissolution. And that, of course, makes perfect sense because your sense of where you are, where your persona is defined in your posterior cingulate cortex, because that's where your relationship with the outside world is encoded. So this was very compelling, and it's clearly a neuronal effect of psychedelics. But the time resolution of MEG is such that we're able then to do a more sophisticated analysis. And this was work by Rosalind Franklin and Carl Friston at UCL. They'd come up with a simple model of the cortex, with two pyramidal cells, two inhibitory neurons, which they believed could explain the oscillatory profile of the cortex. And we, using psilocybin, which produced powerful disruption, here's alpha rhythm completely disrupted by psilocybin, they were able to then test the four theories that they had and see which of them was most likely to be the site of action of psilocybin. And they showed here that, in fact, it's the layer 5 pyramidal cells, the target of psilocybin to produce these changes. And I think that's the first time in human science that someone has actually been able to say categorically that a drug effect is mediated by a specific subtype of neurons in the brain, the layer 5 pyramidal cells. We've subsequently gone on to ask the question, is DMT the same? And it certainly is. Here you see the profound disruption of power, particularly in the alpha range, produced by an injection of DMT. The DMT goes into the brain, disrupts activity, and as it comes out, the activity settles back to normal. So these drugs all produce this powerful disruption, which we call an entropic brain. The brain becomes much less organized. And that disorganization then allows the increased communication. And I've come up with the idea that maybe there are two forms of consciousness now. There's the classic form of consciousness, which you're all experiencing at present, which is driven by glutamate. Glutamate is responsible for arousal, attention, memory, and the parcellation of data, the phenomenal precision that your brain has in learning new information. You've learned vast amounts of new stuff, very discrete pockets of information, packets of information already from my talk today. But under psychedelics, by stimulating the 2A receptor, they change the nature of the memory. They don't impair the memory. People still can talk vividly decades later about their trips, but they alter the content. They change the valence. Often it's pleasurable, sometimes it's not, sometimes it's challenging. They change the meaning, and they produce integration. They allow people to make sense of things rather than facts. They make understanding. And this, I think, is a very kind of useful way of conceptualizing what psychedelics do. So if you want to read more, there's a little review we put in Cell a couple of years ago, which takes you from the neuron to the entropy to the connectivity. But also, what's interesting about psychedelic research in humans is it's now spawned back translation into animals. And animal work shows that psychedelics have quite a powerful neuroplastic effect, which is basically shown schematically here. Following administration of psychedelics, you can show increased dendritic arborization and increased synaptic growth. And that, we think, is one of the reasons why psychedelics can produce such enduring changes in the way people think about things, because they facilitate the laying down of new insights, new memories, new attitudes, etc., through plasticity. I just want to briefly show how different psychedelics are to other drugs using MEG. So over here, I've shown you this already. I've shown you psilocybin disrupting the power of synchronicity. And the same is true of LSD, and the same is true of ketamine. These drugs completely desynchronize brain activity, and that is almost certainly why they work. But over here, you have drugs that do the opposite. You have drugs which potentiate GABA, particularly extrasynaptic GABA, and they produce massive increases in synchronicity. The red is greater power in these low frequency bands. And you can see, there are really clear differences between all these different drugs in terms of the MEG profile. And here's alcohol, which is some kind of hybrid of the GABA and glutamate drug. And then finally, I want to explain how this field, we've developed this field, and how we've moved into the treatment of depression. I want to emphasize this is something we didn't expect to do. The only reason we started studying psychedelics in depression was because of the findings we made when we did the brain imaging. But of course, there's a lot of reason to study depression. It's a huge cause of disability. Treatments are not very effective, and often it starts in adolescence and lasts for people's lives. So it's very disabling. So why did we study depression? Well, there were two reasons. The first was that our discovery that psilocybin switches off the brain pointed to a region being switched off called the subgenual cingulate cortex in the medial prefrontal cortex, which we knew from other people's work was switched off by antidepressants. We know that SSRIs and CBT, over time, as people get better, this brain region becomes less active. Even placebo, if you get better on placebo, activity in the subgenual goes down. And in fact, that is why Helen Mayberg went away and did deep brain stimulation in this region to switch it off, and actually showed if you switch it off, you lift depression. And that's because this is the part of the brain which drives many of the negative thinking and thoughts and ruminations of depression. And ketamine does the same. So we thought, well, if we can switch this off, maybe we can immediately lift depression in the same way as switching it off with deep brain stimulation lifts depression. And the other reason was because of the discovery that that control network, which is switched off in the brain, is actually now called the default mode network. And it's the network in which you embed your thinking about yourself. And the default mode network is a network which is the anterior posterior cingulate, these lateral parietal regions. And this is where you encode your sense of self. And if you're sitting quietly, not listening to anything, not seeing anything, and just thinking about yourself in a scanner, then these regions are active. And all the other regions, the visual and motor cortex, etc., are switched off. And this is where, really your sense of self is embedded. And psychedelics completely disrupt this. Default mode disappears, which is why you often feel that you're not even in this present universe. You might go somewhere else. And that disruption turned out to be very relevant to depression, because about the same time as we were doing our work, this group in the States had come up with this remarkable discovery of hyperconnectivity of the default mode network in depression. And you see much more of the brain is connected when people are just reflecting on themselves in the depressed people, the major depressive disorder, than the controls. And it turns out that the greater the connectivity of this network, the greater the rumination. And that makes perfect sense, because that's what depression is. Depression is a disorder in which people think repetitively about themselves in a very negative way. And the driver for this hyperconnectivity is the subgenual cingulate. So we thought, wow, if we can disrupt the default mode, maybe we can disrupt depression. And there was one other study that had been done in Johns Hopkins by Roland Griffiths and his team, and they'd taken normal middle-aged people who were just interested in thinking more about life and themselves, and they gave them psilocybin in one session. And many of these said it was a powerful, life-changing experience, which was sort of a bit similar to an antidepressant effect. So we thought, well, can we study depression? And in 2012, the British government had a call for new treatments of depression. And we actually got this awarded, the only time we've ever gotten any money from the British government to do this research. Getting the grant was the easiest bit. It took 3 iterations to get the ethics committee to allow us to do the study. And they said, no, it's too dangerous. Depressed people might die if you give them psilocybin. Maybe they go mad. And in the end, we weren't allowed to do a controlled trial. We had to do a safety study. And so the clinical data I show you were secondary rather than primary outcomes. But you'd be pleased to know that no one did die. But that was easy in getting the drug. Only one place in the world would make it for us, because it's such a controlled drug under the UN conventions. It took 30 months to get the permissions to make it and send it, and another 2 months to get the British regulators to let us approve it. So it took 32 months out of a 36 months grant to do this research, all because magic mushroom juice is treated as the most dangerous drug in the world. It's a Schedule 1 drug under the UN conventions. It's treated as more dangerous than heroin or fentanyl. And the costs were enormous. But we did it, and the results were dramatic. So we took people who had failed on at least 2 antidepressants. They'd all failed on CBT. Some had failed on over 10 antidepressants. We gave them a 25 milligram dose of psilocybin, and dramatically, most of them improved. Some of them became completely cured. Under 5 is a complete remission of depression. You can see the effect sizes here, Cohen's D, huge changes, the most powerful impact of any single treatment on depression there's ever been. Absolutely remarkable. And some of them are still well now, not many, about 3 or 4 are still well. For most, they had a benefit that lasted weeks or months, but eventually the depression crept back. And that tells us something fundamental about depression. It's almost like it becomes the kind of base state of the brain, like a habit in the brain, which has to be, maybe we have to keep suppressing it to allow people to escape. But at least for a period, these people, most of them were extremely grateful and much happier than they were before. And how does it work? Well, I'll go back to this image I showed you before. We think during the psychedelic trip, people understand their problems and can make up, come up with new ways of thinking about them, and also come to deal with them, to get closure. And I think it's that liberating the brain allows them to liberate themselves from the depression, which has been kind of forced into the brain by this brain becoming over efficient at maintaining thought processes. And subsequently, we asked a different question. We've just published this paper this last year. It was a comparison of psilocybin, this time, 2 doses, to see if we could get a more prolonged effect, and escitalopram. Escitalopram is a very powerful, safe SSRI. So we compared the two, but to get clinical equipoise, it was difficult. The way we did it was to tell everyone that they would get psilocybin. But half of them got a high dose and half of them got a low dose. The ones that got the high dose, they went then on to placebo, and the ones that got the low dose went on to escitalopram, 10 milligrams and then 20 milligrams, and the output was at 6 weeks. And here's what we found. The study wasn't powered to look at clinical outcomes, but you can see the green bars show significant benefits for psilocybin over escitalopram. You see, in fact, on every measure but one, psilocybin was better than escitalopram, and the remission rates were hugely better. So the magnitude of the improvement was much greater on the psilocybin. And you can see that here. We had 4 measures of depression, and on all of them, psilocybin at 6 weeks was twice as good, sometimes 3 or 4 times as good as escitalopram. And since remission is really one of the key goals of treatment, this was very pleasing. Now, we're not the only people that have done this research with psilocybin in depression. There have been studies looking at people who've got anxiety and depression as a result of having an end-of-life diagnosis like terminal cancer. There's a recent study published last year from Johns Hopkins looking at people with depression who were randomized either to stay on a waiting list, and they didn't do very well, or go on to psilocybin, and they did well. There have been several studies of ayahuasca for depression done in Brazil. But there have also been studies using psilocybin for helping people stop smoking and stop drinking. And also we're now doing studies in anorexia and OCD and pain syndromes. And in fact, as a result of our depression study, the first depression study, there are 40 companies now studying psychedelics and depression. So it's become an enormously interesting area and a busy area. You might say, well, what have all these things got in common? And the answer is they're all internalizing disorders: addiction, smoking, depression, OCD, anorexia. They're all disorders in which people have thought processes which they actually often know are wrong, but they can't disengage from them. And they're all about themselves and about their inability to effect change, etc. So these ruminative disorders, I think, are probably due to over connectivity in the brain, and disrupting it with psychedelics may liberate people from that. So this is my current theory how psychedelics work. Go back to the visual system. As I showed you, you can disrupt the processing of visual reconstruction, but you can also disrupt the way in which the brain decides what it wants to see. And this, I'm going to use this quote of Blake that I showed you before, that man sees things through the chinks of his cavern, and that man needs to basically open the doors of perception because they're normally pretty closed. But for most of us, when we look through the chinks of our cavern, what we see at least is a blue sky and white clouds. It's relatively positive. But for depressed people, what they see is a grey, miserable world. And for addicts, they see their love objects, the bottle or the syringe or the white powder. And that's because the brain constrains them to see those things. And there's another wonderful quote from Blake. He argued that humans had mind-forged manacles which stopped them behaving in appropriate, rational ways. And I think psychedelics break these manacles. I actually think the manacles might start in the mind, they end up being in the brain, but psychedelics can break them and allow people to escape from these very damaging thinking cognitive processes. Here's a bit of new data, not yet published, it's coming out in Nature Medicine next month. And it shows that the increased connectivity that we found during the psychedelic trip, this increased connectivity is defined by a reduction in modularity. Modularity is the isolation of brain connectivity. So before psychedelics, the brain is more modular. After psychedelics, it's less modular, more connected. That is what we saw during the trip, but we also saw it after the trip. We saw the day after the trip that depressed people had more connected brains, and this increased connectivity predicted outcome. So this is fascinating because it suggests that what we're actually doing is changing the brain in the way we predicted. It makes the brain more flexible, more able to adapt, to change the way it's thinking. And here you see the default mode is over-connected before the trip and under-connected after the trip. It becomes, sorry, let me go back. I didn't make that clear. After the trip, the default mode is less connected to itself and more connected to the executive network and to the salience network. So this increased connectivity is a manifestation of the ability of the brain to escape from its internalizing thinking. And I'm going to finish now with this quote from the great philosopher and playwright George Bernard Shaw. Those who cannot change their minds cannot change anything. And I think what I've shown you, and I've learned myself, is that psychedelic treatment has changed the minds of our patients, almost certainly because it's changed their brains. And I just hope that this research can change the public and politicians' minds about these drugs and hopefully bring them back into medical practice, because 50 years is a very long time not to have them when they're so powerful, and they give up hope that other drugs have not been able to give to our patients. I'll finish now. I'll thank the funders and particularly this family, the Mosley family. This young man in his 20s committed suicide in a depressive episode, and his family gave us his legacy to do that research on the brain mechanisms of psilocybin versus escitalopram, because they want to bring justice, the scales of justice, back into to thinking about drugs such as psychedelics. So I'll stop now and I will take questions. Thank you.
Thank you, Professor, for a remarkable talk. For sure, it gave us a different angle for the psychedelic use in neuroscience issues, also from the historical side. So we have a few questions from our audience. So first one, do you think that psilocybin can be a solution to a disease such as PTSD and major depression, where we can observe changes in the connectome?
Yes. So, did you say DMT? What was the first part of the question?
First, I said psilocybin.
Psilocybin.
Yes.
Yes. So it clearly works in depression. There's no question it works. There are now 6 or 7 studies showing it works in depression. PTSD is interesting. When I started this research, 10 years ago, with the first started the depression study, I was skeptical about it having value in PTSD. I was scared that it might make people more anxious. And there was also work showing that MDMA was useful for PTSD because it calmed emotions, whereas psychedelics can enhance emotions initially. But now I'm learning that many, particularly veterans, Army veterans, are going for ayahuasca retreats. And also some of them are going for mushroom retreats in countries like Holland and Spain. And they are finding it does help with the PTSD. So yeah, I'm much more positive now that it may be that psychedelics can help in PTSD as well. Again, probably through breaking the ruminative memories that actually perpetuate the disorder, but it hasn't been formally studied yet.
Okay, thank you. Interesting. So the second thing our audience is interested in is taking psychedelics taxing for the brain, or is the entropic brain a product of relaxation?
Yeah, it's quite an interesting question. It's not simple relaxation in the sense that I showed you the sedative drugs, which can't put you to sleep, the GABA drugs, they don't produce entropy, they produce synchrony. So it's not simple lower level of consciousness, if you see what I mean. But on the other hand, there is work done with intense transcendental meditation. And the transcendental state is very similar to the psychedelic state in the sense that the default mode network becomes disengaged. So if you can get into that state of completely detaching yourself from the world, you're probably ending up in a state which is similar to psychedelics, perhaps with less of the hallucinations. And that may explain why mindfulness, which can help conditions like depression, it can disengage you from the repetitive thinking, but it's not simple relaxation, no.
Okay, thank you. And the next question, could you elaborate on training OCD with psychedelics? Wouldn't it fuel one's obsession?
Yes, well, so it's again a very interesting question. So our thinking is this: our thinking is that OCD is a disorder where people have thoughts they know are ridiculous. I mean, you know, they know that there's no need to wash their hands. They know they're not contaminated, but they feel they're contaminated. And OCD was the first disorder where it was absolutely clear there was an aberrant hyperactive circuit in the brain, which is orbitofrontal head of caudate circuit. So our thinking was, you know, if we could break down those circuits, at least transiently, people would escape from the OCD during the trip, and they might then understand they could escape from the trip, and they might actually not reset. It might be like you shake up a snow globe and it all settles down differently. So we put it out to our patients, and they said no, they were too scared to lose control, you know, because of course a lot of OCD behavior is about keeping control. So then we said, look, suppose we gave you a lower dose that relaxed your thinking processes but didn't actually cause a trip. Would you be prepared to try that, this psycholytic therapy? And they said they'd try it. So we're in the process of doing that. And the way it's going to work is that they're going to have this lower dose of psychedelic, a 10 milligram dose of psilocybin. And we're going to see if during the trip, we can help them engage better in exposure therapy, get them to do the things, to think about the things they don't want to do, that they're resisting normally. So, but your question's quite right. I mean, it is conceivable that, you know, it's conceivable, I suppose, if things go badly, it might get worse. And we've seen that with OCD in the past, people doing flooding therapy, flooding exposure, people often got worse if they couldn't cope with it. So it is quite a sensitive area, and we're going to take it very carefully. And we've got experienced therapists working with us.
Yeah, all right. And there is a question about hallucinogen, a persisting perception disorder.
Yes.
Why after a trip we sometimes still can see color pattern and fractals in normal size? So
yes, that's a really interesting question. And the answer is we don't know. And it is possible, well, given that, you know, these neuroplasticity changes occur, it's possible that some little bit of rewiring goes on in the visual cortex. All I can say is that this condition, HPPD, is there's a lot written about it. It's not very well studied. So we've now done, I think we've done several hundred administrations of psychedelics, sometimes several times to people. We've never come across a case, but of course it does happen. But I think the big question about HPPD, that as a psychiatrist and a psychopharmacologist, I've been referred people with this problem. Mostly it doesn't affect even their visual acuity. Mostly it's a phenomenon, a bit like you might have a twitch in your ear or your toe. You have a kind of twitch in your eye. The biggest problem is that many people think they have damaged their brain as a result, and they see it as kind of brain damage. So it's the anxiety about the experience, which is usually the biggest problem, not the actual disabling effect, because very rarely does it actually disable them.
Okay, thank you. Interesting. And another issue, rumination is frequently observed in more neurotic patients. Can psilocybin influence as well changes of personality traits? Would you expect the temporary changes or more permanent ones?
Well, there's no question psychedelics change personality traits. That's been well established for decades. Yes, people become more open. They become more interested in other people. They become more connected with the world. I don't know if, I mean, there's some of the people who are pioneers, the leaders of Extinction Rebellion came to it having had a psychedelic experience, realizing actually we were more connected with nature than we used to think, and they used to think, and that nature needed to be supported by humans. So personality does change, and often the changes are very long lasting. Yeah, there's no doubt about that. Whether you can use psychedelics to change what are called personality disorders, like borderline personality, that's much more questionable. I'm not sure you can. There's no evidence at present. You can change fundamental personality behaviors like that, but it hasn't properly been studied.
Okay, thank you. That's all of our questions. So thank you once again, Professor. It was really interesting.
It's been a pleasure. I'm sorry again about the mistake with the time, but I hope most of you managed to stay on, and I appreciate the invitation. Thank you very much.
Yeah, we're really happy to have you here.
Enjoy the rest of your conference, and I'll say goodbye. Thank you.
Thank you.
Bye-bye. Bye.